Thank you all for coming. It is my pleasure to be able to present the work I have been doing for my PhD thesis. The title of my thesis is Transcription Factor Expression Patterns in Developing Vestibulospinal Neurons. To help you digest this title a little bit, I can tell you that we are going to talk about vestibulospinal neurons, which is a type of brain cell that is involved in controlling movement. We are also going to talk about what happens as these neurons are generated during embryonic development. And we are going to talk about how a group of genes called transcription factors are involved with controlling this process. So let's start with vestibulospinal neurons. Vestibulospinal neurons are a type of neuron located in the brainstem of all vertebrate animals. There are in fact different types of vestibulospinal neurons. One of them sends their signals through long arms, or axons as we call them, all the way down to the bottom of the spinal cord. We call this type LVST neuron because their axons run in the lateral vestibulospinal tract. Another type first sends their axons across to the other side of the brainstem and then down only to the top of the spinal cord. We call this type CMVST neuron because their axons run in the contralateral medial vestibulospinal tract. Both the CMVST and LVST neurons contact and activate motor neurons in the spinal cord. These are neurons that send their axons to muscles and can activate muscle contraction, leading to movement. But there are many other types of neurons in the brain that send axons to the spinal cord and activate motor neurons. What makes vestibulospinal neurons so special? Well, this has to do with the sensory input that activates these neurons. You see, just behind our ears, on both sides of our skull, we have something called the vestibular labyrinth. On both sides, this organ can sense head rotation and head translation. Special neurons transmit this information into the brainstem, and they can activate the vestibulospinal neurons when the head moves. But the vestibulospinal neurons are smart. They will not be activated by voluntary movements. It is primarily unexpected movements that will activate the vestibulospinal neurons, and in turn the motor neurons, so that the body can make corrective movements. This is called the vestibulospinal reflex, which is important to help all vertebrate animals maintain upright posture. And it is from the special sensory input the groups of the vestibulospinal neurons get their names. Here we see an example of some goats experiencing lots of unexpected movements. Yet they have no trouble standing upright thanks to the vestibulospinal neurons. So, the vestibulospinal neurons appeared early in vertebrate evolution, and all vertebrates are born with these neurons. But how is it possible to be born with so many different types and different complicated types of neurons? This is partly because during embryonic development, the undeveloped future brain, shown here, is exposed to molecules that divides the brain into several territories with different gene expression. And differences in gene expression leads to differences in neuron form and function. By combining two axes of signals, even further territories can be generated. Here we see two real pictures of how the spinal cord looks like during development. The territories are most visible when we investigate a group of genes called transcription factors. The coloring technique used here is called immunohistofluorescence, which you will see more of later. Transcription factors or TFs, are a class of proteins that are very important in controlling a cell's function because they directly influence other genes. But what about vestibulospinal neurons? If animals are born with vestibulospinal neurons, does this mean that they express specific TFs during development? And what about LVST versus CMST neurons? Are there differences between these? And since vestibulospinal neurons are so special, is there a unique set of transcription factors, or TFs, expressed in these cells which is found nowhere else? And what if we compare mouse and chicken? Will we find the same result? And how can we effectively and efficiently use immunohistofluorescence technique to answer these questions? To start investigating these questions, I dissected out brains from mouse and chicken embryos. Then, I injected a fluorescent compound into one side of the spinal cord such that the LVST and the CMST neurons become labeled. This allowed me to collect samples of purified vestibulospinal neurons and use the RNA sequencing method to check which of the approximately 2,000 different transcription factors they express. With this technique, I would get expression levels for all of them, ranging from no expression at all to very high levels. But many TFs are expressed in all cells, and I was not interested in such unspecific expression. I therefore compared my samples with cells from other parts of the brain. To visualize all 2,000 data points in the mouse, I plotted them with the LVST-enriched TFs along the x-axis and the CMST-enriched TFs along the y-axis. This showed the presence of 4 categories of TF expression patterns. Those with low expression in all vestibulospinal neurons, those with high expression only in LVST neurons, those with high expression in CMST neurons, and those with high expression in both LVST and CMST neurons. When I compared this with the chicken data, it was clear that expression patterns were highly similar. For example, FOX2B was the most specific LVST TF, and LHX1 was the most specific CMST TF, and MAF was highly specific for all vestibulospinal neurons, in similar patterns across the mouse and chicken. RNA sequencing on groups of neurons is very good, but it has one weakness. It cannot tell you if all or just some of the neurons in your sample express a gene. That's why I wanted to use immunohistofluorescence and test the most specific TFs against labeled vestibulospinal neurons. With this technique, I could investigate if only some of the neurons express the TF, as is the case here, or if all of the neurons express the TF, as is the case here. I could also see if the gene is expressed in cells that are not vestibulospinal cells, such as these TFs that are expressed in non-vestibulospinal cells. This analysis showed that on a cell-by-cell resolution, that FOX2B is expressed in all LVST neurons and never in CMST neurons, whereas LHX15 is expressed in all CMST neurons and never in LVST neurons. MAF and ESRG were expressed in nearly all vestibulospinal cells, regardless of type. The rest of the tested TFs could be found in subpopulations of either the CMST group or the LVST group. When I did the same analysis in chicken, I found that the results were overall highly similar. For example, both FOX2B and LHX15 show the same specific expression pattern as in mouse. But as I showed you before, none of these TFs are unique to vestibulospinal neurons and are found in other neurons also. But what if we combine 4 TFs at the same time? Are there any other neurons in the entire nervous system that express these TFs simultaneously, or is it unique to the LVST in this case? To find out, I performed 4-color immunohistofluorescence and compared it to labeling of the LVST neurons. Here you see the labeled LVST neurons, and here you see 3 of the 4 TFs that have been labeled. The white shows where all 3 overlap. This analysis showed that in the entire mouse central nervous system, this TF signature of 4 TFs was unique to a small area of the brainstem. Except for a small group of unidentified neurons, the only cells with this signature were the LVST neurons. This was a rather remarkable result, and I don't think there are many other TF signatures that target such a specific group of neurons. But could we also find a TF signature for the CMST group? For the CMST group, the finding was a little bit different. You see, the brainstem is divided into rhombomeres during development, which have different gene expression patterns. It is known from previous studies that LVST neurons all come from the R4 part of the brainstem, while the CMST neurons come from both the R4 and the R5 part of the brainstem. I chose to focus on the larger R5 CMST group when looking for TF signatures. Again, I selected 4 TFs and checked the entire mouse central nervous system for cells matching this signature. And once again, using a combination of 4 transcription factors, this TF signature was also unique to a small area in the brainstem. And similar to the LVST signature, except for a small group of unidentified neurons, the only neurons with this signature were the R5 CMST neurons. To summarize the results I have presented so far, first, we found TFs that distinguish the LVST and the CMST group perfectly, using the FOX2B and LHX15 TFs. We also found several TFs that define subgroups within the vestibulospinal system. And we saw that the patterns we observed were evolutionarily conserved between mouse and chicken, indicating it might be true for a broader vertebrate line. And we found some 4-membered transcription factor signatures for the LVST and the R5 CMST group, which can prove valuable in the future. These results have been published in the neuroscience journal eNeuro. I will now talk about my second paper. So the second part of my talk will focus on the method that I developed to analyze immunohistofluorescence data. So I will go through how I generated images such as these and how I analyze the data using this new method. So the first thing I did after dissecting out the brain and labeling the vestibulospinal neurons in red is to cut the brain into really thin sections using a very precise knife, and then transfer these brain sections onto glass microscopy slides. Then this glass microscopy slide with the tissue and the labeled neurons could be put under a microscope connected to a camera and to a computer. And then I could take picture of my vestibulospinal neurons. but I was interested in more than just the picture of my neurons. I wanted more information about the transcription factors. But I needed to differentiate between the vestibulospinal neurons and other cells. Therefore, I added a compound that lights up all of the cells in the tissue piece. And this includes not just the vestibulospinal neurons, but also other cells and other neurons. And this particular compound actually binds to the DNA, which means that it is the nucleus of the cells where the DNA is that is being marked with this compound. And this is then something I also could take an image of. But my interest was in finding out which of the vestibulospinal cells has a particular transcription factor. So let's imagine, for example, that these cells here have the transcription factor FOX2B inside of their nucleus, because it is within the nucleus the transcription factors usually are. So I needed to have a compound that can find and light up only these nuclei. And for this, I could use something called FOX2B antibodies. Antibodies can be generated in a way that they bind only to their target transcription factor or any other gene for that matter, or protein. So now, in this experiment, I had labeled also the nuclei that have FOX2B in them. And then I could take a picture also of this. Then I needed a way to analyze this data and to find out which vestibulospinal cells expressed and contained the FOX2B gene protein. So I combined these pictures and then I could analyze exactly this. But hold on, there is something wrong here in this picture. These vestibulospinal cells appear to have the FOX2B signal overlapping with it. But when we look at the original data, we see that this is not the case. The vestibulospinal cell does not have FOX2B. So this signal is coming from underneath the cell. But this type of imaging cannot distinguish between these two cases, because this type of microscope just takes a top-down view and you get this 2D projection of something that is actually 3D. And what we have here is something we call the false positive signal, because we do not take into account that this tissue also has a depth. So what could we do to avoid this problem? Well, one thing we could do is to make really thin sections of the brain. Then we would only get pictures from a single thin plane and we would not get overlapping cells. However, that is extremely difficult to do because these sections are already very, very thin. Instead, we can use something called a confocal microscope, which is an advanced microscope that is able to do something called optical sectioning. Optical sectioning is where the microscope is able to collect light only from restricted thicknesses and depths of the tissue. And here you see, for example, how this microscope is taking an image from only the bottom part of this tissue. And it can also take a picture from the top part of the tissue, or even many thinner parts. And here, in this top part version of the confocal images, you see that this FOX2B signal from this lower right corner here has been removed from the FOX2B image. And when we analyze this data, we call this a set stack. And when we analyze this data, we see that the false positive signal is avoided. So I made sure to always use a confocal microscope when doing my imaging experiments. However, there is one problem that cannot be fixed by using confocal, and that is because of something that happens during the physical sectioning of the brain tissue. This sometimes leaves some cells cut in half, and then often the nucleus is stuck in the other half that is not included in the section. And now if we take a picture of this, it will look like this in the different channels. And if we overlay them, these two cells here actually appear to not have any FOX2B signal in them or any nucleus signal. But when we look at the original data, we see that this cell actually has FOX2B in it. It is just not included in this section. So this is actually a false negative signal. And what can we do to eliminate this false negative? Well, we have to realize that it is only if we can see the nucleus of a cell, such as in this case, or in this case, here and here, that we can actually know whether these cells has FOX2B or not. So if we can't see the nucleus, it's impossible to know, because it might be in a FOX2B in another cell, in an adjacent section, or it might not be. So we have to exclude this from our data, and hence we avoid false negative counting. So here you see an example of how I actually analyze the data using these two principles. And first I find the neuron, this one, that I'm interested in, and I scroll to the correct position, and then I see it has a nucleus. Then I check if it has a transcription factor. Yes, it does, so I mark it. Then I go to the next neuron. I choose this one. I find out that it has a nucleus. Good. And then I check for transcription factor, which it does not have. So I mark it as a negative cell. And now I can be sure that I'm counting correctly. However, I had to count tens of thousands of cells. So this took a very long time, and I really wanted to find a way to do this faster. So I realized that I was not really interested in the signal from the vestibulospinal neurons or in the nuclear signal. I was only interested in the signals where they overlapped. So I processed using a computer the pictures and removed everything except for where these two signals overlap or co-localize. And then I could use this to compare with the transcription factor signal. However, I could not simply overlay the picture, because then all the green signals would be hidden under the white signals. So I came up with the idea of converting this white signal into outlines. So these showed the nuclei of all the vestibulospinal cells, and now I could overlay the transcription factor image on this image. And this allowed me to easily and quickly check which vestibulospinal cells actually expressed the transcription factor in question. And I could easily see which ones did not express the transcription factor. And now I only had to look at one picture instead of scrolling back and forth between three pictures. So this was a huge advantage. And I even found a way to combine the complete Z-stack of images into a single image so that I didn't have to scroll through anything. And this saved also a lot of time. And with these types of clean images, I could even use automatic detection algorithms to mark automatically for me the cells that were positive and the cells that were negative for a transcription factor. And I could be confident in that I didn't have any false positives and false negatives. So here you see an example of a complex image that has been transformed into this more simpler image that is fast and quick to analyze and doesn't have any of the problems I have described. So I wanted to share this technique that I thought was very useful with the world. So I decided to convert this into a flexible algorithm and to make a user-friendly menu where you can simply click and choose what kind of experiment you have, and the program will automatically find a way to put this data together and in a way that hopefully avoids these false negatives and false positives. And this has been made as a plugin in the program ImageJ. And furthermore, it also comes with a plugin that allows you to use this automatic detection and something that can speed up counting very much, and something that helps to keep the data organized in Excel sheets. And it even has the ability to combine data from multiple images so that you can create a 3D reconstruction that is color-coded according to whether the cells are positive or negative for a transcription factor. So we thought that this was something that not only neuroscientists could use, but also other types of biologists, and maybe even outside of biology. So this has been submitted to the open access journal Scientific Reports, which is a broad type of journal. So to summarize, we found a method that could be very efficient to analyze confocal data with if you have a lot of different labels, fluorescent labels, and especially if you analyze transcription factors that are only staying in the nucleus and avoid certain types of false negatives and false positives. And we also designed a complete toolbox that allow you to quickly count, organize, and make 3D reconstructions and visualizations of this. So for both of my papers and my whole PhD, I want to acknowledge the people that has helped me with this, which has been many people in the lab, and of course my supervisor Joel. And then I might include a slide about what this information is good for. Thank you.